Well, good morning, everybody. It's nice to be back in Texas, but I flew in and got sick, so please excuse me if I sound a little hairy. I thought I'd better give you a little bit of background. I started off with just putting James Madison University, and I figured everybody would be going, what the heck is she talking about Texas coastal waters when she's in Virginia? So I was in Corpus Christi for a fairly long time until last year. And I just wanted to talk a little bit about some of the early work we did with BST before talking about the library-independent markers. We started a little bit differently because we're on the coast, you know, Corpus is on the water, and one of the issues that we hit on really early was most of the waters we were working with were on the 303D list, TMDLs somewhere lurking, and so there was, at that time, back a few years, a lot of concern about relating the BST to fecal indicators. And so when we're working with coastal waters, either fecal coliforms for shellfish harvesting waters or Enterococcus are used generally as the fecal indicator. And so we moved away from E. coli. So George and Terry are doing all the inland Texas E. coli work. And we started using E. coli we sort of justified it because, you know, E. coli is a subgroup of fecal coliforms, and so we were working with shellfish harvesting waters in Copano Bay, Cedar Lakes, and some of those areas. And we've sort of recently moved to focusing on Enterococcus because we're working with tidal watersheds, and while E. coli is recommended for freshwater, Enterococcus for marine water, there's also that overlap that Enterococcus is an approved indicator for freshwater as well. So in order not to have tidal watersheds where part of the river is freshwater and part of it is tidally influenced, and at what point do you change from E. coli to Enterococcus and you have data of different indicators, for those we've been using Enterococcus both for monitoring and for source tracking. So when we first started, we were asked to look at some of the initial BST methods, and we started off actually with pulse field gel electrophoresis, which, if you remember George's talk yesterday, no, not George, Elizabeth, sorry. She had the thing about the sex estate library and the PFGE being way over here as the most discriminatory and antibiotic resistance being over here as the least discriminatory. We started off with ARA and PFGE, and fairly rapidly decided PFGE, as Elizabeth said, is, yes, it's really, really discriminatory, and it's really, really too discriminatory for source tracking in coastal waters at least. So we moved to working with initially antibiotic resistance analysis, a little bit differently from some of the other groups. We went with the Kirby-Bauer method, which allows us to use image analysis to get zones of readings, which can be put straight into spreadsheets. And with the sort of move towards the toolbox approach with the Texas State Task Force, we started looking at adding other methods, and we added carbon source utilization, which is not mentioned in very many places and not very many people use it, but we've actually had a lot of success with it as a phenotypic library-dependent method. You get profiles of 95 carbon source utilizations for each bacterium, and then we add antibiotic resistance on there, and we end up with this big profile of 120 numbers that we use to compare. We've also started looking at some different statistical methods. Almost all of the antibiotic resistance analysis, source tracking has been analyzed using discriminant analysis. We've started using another tool called random forests, which my statistical friend can tell you a lot more about than I can, but we've had pretty good success with that. So we've been sort of evolving in parallel, but sort of a little differently because we've been dealing with coastal watersheds rather than inland freshwater. So a few years ago, we sort of realized we needed to start adding to the toolbox, and again, coming back to the task force recommendations of looking at some library-independent methods in addition to the library-dependent studies we'd been doing. So I'm going to talk about a couple of things, and they are preliminary pieces of studies, I guess is the best way to put it, sort of cobbled together from different pieces of funding. So it's not a complete story at this point. It's some preliminary, this is where we're at, and some of the potentials to continue. So one of the first things that happened was Jody Harwood had a PhD student who graduated and came and worked in my lab as a postdoc. And so that was great for us because they'd been working with a lot of library-independent markers, and she was able to bring that expertise to the lab for us. And in conjunction with that, Jody and two other PIs had some Gulf of Mexico Alliance money, which I don't know how many of you are familiar with Gulf of Mexico Alliance, but it's a whole different set of funding across the Gulf states. And because we're coastal, we were able to link in with them. And so Jody's group had decided to look at some different independent markers, do a whole serious set of validation, just Terry was talking about the need for validation in different areas. So they started off, came down to 3 markers and validated them, and then wanted to do some field testing in different states. And so we came in with Jody to try testing those markers in our coastal waters. And we looked at both marine waters and freshwater. And then because we were looking, we've been working with Enterococcus, we also wanted to start looking at trying out the Esp marker, which I'll talk about a little bit more, which we haven't had anybody talk about here yet. And again, we used that both from marine water and freshwater studies. So you've all heard this before, so we'll go through some of this fairly quickly. Library-independent methods, you don't need to go back to this library and form this big group of different animal source isolates of E. coli or Enterococcus or whatever you're planning to do. They actually just are identifying a particular target. And there's a whole range of them out there. You've heard about a lot of them already. What Jody's group came down to was 3 human-specific markers. And so all I'm going to talk about, all we've worked on so far has been human-specific, because for a lot of watersheds, that's what the first question is. And again, comes back to the importance of knowing what's the question? What do you really want to find out? And it can be very different for different watersheds. For beaches, human, non-human is one of the first steps. And so the 3 human-specific markers, Bacteroides species, Methanobrevibacter, smithii, and human polyomaviruses. And again, these are, I'll talk a little bit more about these, they're very diverse markers. They've been evaluated for sensitivity and specificity with Jody and her group. And the use of 3 sort of comes back to the same toolbox approach. It's better to have 3 different ones. You may not get all of them being detected at the same time, which you'll see is what happened with us. So the human-associated Bacteroides, again, just for those of you maybe not so familiar with these, I just wanted to sort of make clear that the 3 markers are very specific, very different groups. So Bacteroides, you've heard a lot about Bacteroides from various speakers. And so this was one of the markers that's been very widely used, both in Texas, in Gulf region, in France, Australia, you name it. It's been worked on very extensively. Methanobrevibacter smithii is actually a member of the Archaea, totally different group of microorganisms. It is found in the digestive tract, and there are primers that can be used for this organism. And then the third one are the polyomaviruses. And again, there's been very little talk about viruses in this meeting. And Jody's group started looking at the polyomaviruses. They're very specific to human feces and very sensitive. So again, the methods we were looking strictly at presence-absence. It was not qPCR. It was just, can we detect them or can we not detect them? We followed protocols that were provided by Harwood and her group. And so everything was standardized in terms of following the protocols they had already established. We did do a lot of initial validations with sewage samples, limits of detection. I'm not going to go through all of that. We used standard primers, again, the ones that had been optimized by that group. And then we, like I said, added, and we had a little bit of separate funding for working with ESP from the General Land Office and Basin Estuaries Program. And this Enterococcus surface protein, or ESP, is a protein that's found in Enterococcus species associated with human intestinal tracts. And it's, again, being used in several different studies, both in Florida and in Australia, a number of different areas, with varying degrees of success. So the ESP gene indicates the presence of human-associated E. faecium, which is one of the species. One of the big differences between working with E. coli as a fecal indicator and Enterococcus is that Enterococcus is a genus level. And so E. coli, you're looking at a species. I mean, there's a zillion different strains, but it's a species. Enterococcus, you have an additional sort of degree of complexity in that you're working with a lot of different species. And we've been doing some other work looking at species of Enterococcus in different animals. Is there any way we can use any of that in terms of source tracking? And the ESP methods, again, very similar. Your membrane filtering, enriching, and then it's a pretty standard PCR methodology. We also, for all of these, we're running fecal indicator levels using Enterococcus following EPA method 1600, very standard. So all the way through all the samples we took that we analyzed for the library independent markers, we also have Enterococcus levels. And again, I'm not going to go through the methodology. I will just mention, you can see here for the ESP, we had the same number of controls for the markers from Florida. There's a lot of controls. I mean, there's a lot of talk about it's not as costly, it's much quicker, it's much easier. I have to tell you, it's not quite as easy and simple as it sounds when you're down on the ground and you're running how many different controls. And when we were doing the 3 markers, we had all these positive, negative controls from the environment, and then positive, negative controls all the way through PCR. There are issues with PCR that can affect the results in terms, particularly in terms of inhibition. There are substances in the water that can inhibit your reactions. And so we have all these extra built-in steps to make sure that, yes, we aren't having inhibition, we are having positive controls, etc. So just sort of when you're down on the ground doing it, it's a little bit more than just, okay, throw it in a tube and you get a result. So for Corpus Christi area, for those of you not from around Texas, it is a fairly large urban area now. Tourism is key, and this has been one of the sensitivities. We've done a lot of work with beaches, and they really don't want to hear too much about impaired beaches because of tourism. We also have some number of different estuaries of national significance. We've got fisheries, a lot of recreational use. And so anytime in this area that you've got some kind of impaired water segment, it's a very high profile case, and we really have to be looking at what's happening. So we're going to just talk about a couple of examples. One of them is Corpus Christi Bay. Before I get to Corpus Christi Bay, sorry, just meant to tell you here, what we were trying to do in these initial studies, and like I said, they were sort of pulled from different areas, was to really look at, could we potentially use these library-independent markers, first off with PCR and then hopefully moving to qPCR. And so we hadn't, nobody had done anything along the coastal areas. In Texas, Florida's really different, Louisiana's really different, waters are different, salinities are different, name it, it's different. And so we wanted to just see, can we use the markers, can we detect the markers in fresh waters and marine waters in our area, and then is there a human source contribution either for the bay sites or for these other creeks? So Corpus Christi Bay, many of you may have remember, or there was quite a bit of publicity about these two parks in Corpus Christi Bay that were being monitored by the Texas Beach Watch program, which we were doing that monitoring, and that they were included in the, actually in the 2008 and in the 2010 303D lists as impaired based on the Beach Watch data. So these two parks, there are 4 sites at Cole and 2 sites at Ropes Park that are routinely monitored through the Beach Watch program. We use the same sites. They both have stormwater outfalls. The one on the right is one from Ropes. It's just right in the middle of the beach area, and you can see there's water coming out or there's not water coming out, and it's all residential area. We're talking about really just a little bit down the bay from downtown. Cole Park is the one on the bottom left. That's the biggest outfall. You see a huge plume comes out after rainfall. It's got a pretty big catchment area, all residential and industrial downtown areas. So the figure on the left is the, so this is Ropes. There's 2 sampling sites. Actually, the storm drain is sort of right about in the middle. There's another storm drain up here. One of the things when we first started way back working along Corpus Christi Bay was to try and find areas where there were no storm drain outfalls. So we had a reference site. It doesn't exist. There's storm drains all the way along the bayfront. Cole Park, we had 4 different areas. Cole Park is an interesting area. It's a park, but there are little beach areas along the park. When you first look at it, you go, it's not even a beach. What are we talking about? But there's a beach here right next to a hotel up here. There's a beach area here and here and here. So for this study, we collected samples for 6 months on a monthly basis, and then we also did some after rainfall. This is one of the things when you start to look at the results, it's not as easy as it looks. How do you base significant rainfall? We had this, 2.5 centimeters, i.e. an inch of rain in 24 hours, or we had a sort of a higher 3 inches of rain over 7 days period prior to. You have to use some kind of number. How that relates to how much water is coming out of the pipes, we don't know. We were not able to get flow data from the city. I don't know if there is flow data. We tried to get samples from the discharges. Some constraints. We were collecting 3 samples per site. We had 6 sites. So we were collecting 18 samples each time we went out. And I'm mentioning that because when you look at the results, it's out of 18 possible sites did we detect the markers. So I'm sorry, this is small. I don't know if you can you read it at all in the back? Yes? Okay. All right. So this summarizes what we found in these marine sites. Oh, sorry. So the ones with the asterisk fall under our definition of rainfall events. But you can see, if you look at the Enterococcus levels, for some of these, yes, the levels were higher, 424 here, but we had a high number which under one event that was not classified as rainfall, and then these 2 gained both rainfall events, and you've got a 10-fold difference in numbers. So there's not a really nice relationship there. Okay, so this is the total sites where we detected 1 or more of the markers out of the 6 sites, okay, 2 at Ropes and 4 at Cole. So you can see some cases we didn't get any 2 sampling events. We didn't detect any human markers, any of the 3 or 4, because we were doing ESB as well. After this rainfall, all 6 sites, we did detect human markers. You can see there's a difference between which markers. We didn't consistently get all of one marker being detected each time. Sometimes we got the polyomaviruses, sometimes we got the H-back, all the way through. Sometimes we got the smithii. Okay? So using multiple markers, you really kind of see, sometimes you detect one, sometimes you detect another. So when we looked at the data, depending, again, how you analyze the data, you can find different things. If we just looked at 3 and 7 day rainfall against detection of a marker, we did get a significant correlation. We also got a correlation with Enterococcus levels. But if you look at it a little differently, which Jody has just submitted a paper with, and she looked at whether or not the Enterococcus level exceeded the standard of 104, and then whether a marker was detected. And doing that, 3 of the labs, she had 5 labs doing this, 3 of the labs did have an association, but our data did not show an association. So again, how you analyze the data, you can really look at different things. This is the same information. Oh, wait a minute, I'm going back again. No wonder it's the same information, sorry. I was going, wait a minute. Okay, so one other thing we started to do, and again, if we'd had more time, more data, could have done a little bit more, was we looked at the effect of the distance from the outfall. So these are distances from the outfall, the 2 at ropes between like 50 and 150, and then the ones at coal, the distance, so this one was the furthest from an outfall, and the percent of times that we had a marker detected. So we were sort of trying to get to, is there human contamination coming out of the storm drains is really where we were trying to get to with this. Okay, so I want to talk a little bit more about the Enterococcus, and then we'll move on to the freshwater. So another little complexity when you start to actually look at the Enterococcus concentrations, what I showed you on those other tables was the average Enterococcus concentration across the 6 sites. We didn't look at it by site. If you start to look at it by site, even in dry weather, you can see there's a lot of difference in Enterococcus levels between the 6 stations. So when you're taking an average, what does it really mean? Should we be looking at the number of markers at each of those sites separately? Because we've got a big difference in Enterococcus levels. This is the ESP data, and I'm sorry, this is out of one of my students' theses. Didn't come. The main thing is, so these are the Enterococcus levels. These are the 6 stations with 3 samples per. These are the Enterococcus levels, and then these are the ESP. And the noticeable thing is there's a whole lot of minuses and no pluses. So we did not detect the ESP marker at all during dry weather, as we classified dry weather, even when we had over 1,000 Enterococcus. Okay. When we looked at the post-rainfall, our definitions of post-rainfall, again, you can see, what I'd like you to show you is the really high levels, we didn't have a lot of rainfall. And again, that's been referred to. It's a little problematic when your life is driven by rainfall and rain doesn't happen. And you've got 6 months of sampling. And so actually, our biggest rainfall was after the end of the study officially. And we just went out there again. And that was when we really had rain. And if you look, these 2 are the Ropes Park sites. These are the coal parks. You can see the Ropes Park, we had higher levels of Enterococcus. And we're talking now reasonably up in the 2,000–3,000. If we look at the ESP results for these are the 2 Ropes sites. And lo and behold, this is where we were able to detect ESP, and we had the highest level of Enterococcus. So is it a detection level? We were kind of getting desperate during the study. It was, well, what's going on here? We have no ESP. We were getting some of the other human markers, no ESP, what's the deal? Do we even have faecium? So we did a little study to see, do we actually have faecium present? And we were able to show that, yes, we did have faecium in those water samples. Now, I have to say this was the one after rainfall when we did get ESP. So for ropes and coal, we did detect human markers. Different markers, different dates after different conditions. Smithii was detected the most frequently. Esp was detected only after rainfall, when we had the heaviest rainfall and the highest Enterococcus levels. So freshwater, very quickly, for the part of Jodie's worksheet, they asked us to also look at freshwater sites. And so the best ones we could find where we knew we had high levels of fecal indicators were wastewater treatment effluents from the Corpus Bay watershed. Again, it was part of another study. When we were collecting the samples, we went ahead and did the markers at the same time. Again, these were associated with Mission and Aransas rivers, which were listed on the 303D list. And we had done a library-dependent study of the Mission and Aransas rivers and found quite a bit of human contribution. And lots of these little wastewater treatment plants. So we got data. We only had one sampling event for these because you probably know a lot of treatment plants are not really happy when you come and try and take samples. We went out with a TCQ and there's this river authority so that we had access to the outflows and were able to get samples. And the results, again, like I say, this was only a one-time. We would love to do it more, but this was what we had. So we were doing fecal coliforms and E. coli and Enterococcus as part of this other study with the River Authority. And here are our markers. Now, if you look at it, it's a little depressing. We have, okay, so no fecal indicators. And for most of these places, it's either really high or it's not. You know, either their treatment has been effective or not. So no indicator, bacteria, or we couldn't detect any. No markers. But Skidmore, no fecal indicators. We detected all three of the markers. Here we have fecal indicators, not really high levels. We didn't get markers. Here, no fecal indicators and no markers. Here we have low levels. We got 2 markers detected. And here we had 2 markers detected. Now when we had pretty high levels of fecal indicators, we did detect all 3 markers.
Is that colonies?
Yes.
CFUs for
yes, they're membrane filtration standard methods.
Why are your fecal, and it's all from the same sample?
Yes.
Why are your fecal numbers lower than?
I'm sorry?
Why are your fecal numbers lower than your?
Oh, got me. I mean, we use standard methods. This is one of those. It's not the same actual piece of water. So, you know, you...
Maybe you get colonies in one sample.
Yeah.
Part of the sample.
It's one of those things that happen.
Was it disinfection at most of these plants chlorine or?
I think so in that. I don't know, but I think so. So, you know, this is a first step. It would be great to do a whole lot more work on it. This was sort of our first what's happening. Okay, the last thing I wanted to just talk about briefly was this ESP study we did. So we didn't do ESP for those freshwater ones because of this time difference and where we had funding. But Oso Creek we've been working on for a number of years. We've worked on it all the way down from the top through down to Oso Bay. It's been listed on the 303D list for a number of years. We did some early work with the Bay, and TCQ wrote up a report, and it's off the list. But the creek is still listed, and we did a lot of intensive monitoring over several years and discovered what we didn't think was going to happen, which was that at the top of the creek, which is through row crops, pasture, out in the boonies, we had really high levels of Enterococcus. We thought we were going to see this nice as we come into Corpus, and you have landfills, and you have treatment plants, and you have residential areas and runoff. We'd see this sort of increasing level of fecal indicator bacteria, and it would start low upstream. That's not what happened. So we've been sort of following the task forcing about the targeted approach. We started off with some routine monitoring, then we did intensive monitoring, we did target monitoring of a bunch of different inflows. found basically fecal bacteria everywhere. Didn't matter. Every inflow we looked at had high levels of fecal bacteria, so we were down to what's happening. And so we wanted to go upstream because we really didn't have an explanation for why did we have these high levels upstream. Now, note that it does receive effluent from Robstown Wastewater Treatment Facility. But we've sampled that, and every now and then there's a little, you'll find some fecal bacteria, but most of the time it stays pretty well. So here's the watershed. So here's Robstown, and we have one site that's literally just down this drainage ditch from where the outflow comes from the treatment plant. Then we have we've got a site here and a site here, which is on this main branch of the creek. We've got two sites over here, which are on a west branch of the creek. So this has been the study area, and the Soil Board has been funding us for several years to really look at this. And so again, the ESP was a very small piece of a much larger study. We were looking at physical sources of contamination. We looked at groundwater, subsurface, runoff from fields, in-stream, sediments, to try and figure out where the bacteria were, where they might be coming from. And then we also did a library-dependent BST study using the composite ARA CSU profiles with Enterococcus. And we just finished that and showed that there was actually very little human contribution. The majority is avian and non-avian wildlife, and there's some livestock contribution. So the ESP we were adding as a sort of an independent evaluation of the human source. So from what we saw, we couldn't see human. From Robstown, we didn't think there was very much. And so this ESP was sort of another independent, are we going to see the same thing? So we collected samples quarterly from those 5 sites in the upper section. And this just sort of summarizes, these are the sites, 18499, ABC, 3 samples per site. The fifth site, we didn't have a lot of rainfall, and it was dry a lot of the time, so I didn't bother showing you. The few sites we didn't ever have ESP. We had ESP detected one time at this one site, 18499, which is upstream. This is the one just down from the treatment plant. We had one detect here and one here, so 2 sampling dates. And then we had several at 2 times at this 18500. But overall, not very much in terms of detection. If we look at it in relation to Enterococcus levels, I just had a few hours to try and help guide you through this. Here we had 8,000 Enterococcus, no ESP. We have much lower Enterococcus, 500. We did get a plus here. We did detect it here with about 500. And down here, we had pluses, detects of ESP with very low numbers of Enterococcus. We were down to 80-something Enterococcus, and we were still able to get a detect for an ESP marker. So the ESP marker was detected at 3 of the sites. One time, 2 samples downstream from the Robstown treatment plant, so that's not unexpected. You'd kind of think you might see it there, just from a few little, no treatment is 100%. And then 18500, we really don't have a good explanation why that it is on the main creek. And we did have some detects, but the majority of the time and the majority of the sites, we did not find ESP. So in summary, as you can see, like I said, it's sort of a work in progress, and where have we got to? The good news is we did detect all of the human-specific markers that we tested in both marine waters and in fresh waters. I would strongly support the toolbox approach. I think it's really important not to depend on one particular method, one marker, one set of markers. If you can do library dependent and independent, I think that's a strength. If you use multiple markers, you have definitely an increased likelihood of detecting human fecal pollution. I think the ESP, even though it was a very small part of the Oso, did kind of agree with what we'd found and what we'd seen from sanitary survey, what we got with the library-dependent study. The beach sites were more complicated. It does seem that there is a human contribution to the contamination at the beach sites. The ESP we only detected after rainfall with high levels of Enterococcus. One just note, not going to comment, but if you look at exceedances based on Enterococcus and when advisories were posted for the beaches, there were no advisories posted based on Enterococcus levels 53% of the time we had human markers detected. So based on these human-specific markers, there was human contamination. Based on Enterococcus levels, it did not exceed the standards, and there was no advisory for the beaches. The ESP, why did we detect it less than the other human-specific markers in marine samples? It could be that there were non-human sources of Enterococcus during dry weather. We haven't been up the pipes. There's been, I know some just talk about biofilms in pipes, rats in sewage systems. We don't have that data. We've tried to get some sampling at the pipes, but haven't been allowed to do that. There are some PCR inhibition issues, maybe just that the level of ESP was below the detection limit. And it may be that our human population is carrying lower concentrations of ESP. Freshwater, like I said, we did find ESP with low levels of Enterococcus, so that's was sort of encouraging. We did find that we were able to use Inhibitex, and it was effective in reducing inhibition. And the absence of ESP when we had high levels of Enterococcus does suggest that we had non-human sources of contamination, which again supported what our other studies for Oso Creek. So lessons learned in future directions. George said, be positive, so okay, being positive. One of the things is, you know, from the beach sites, if you look at it, there's a lot of complexity in the factors that affect your results. And it really takes a lot of untangling from the basic, we went out there and we found a marker, to when did we find the marker? We had issues with rainfall levels. Was there discharge from the pipes or not? How much sampling, how much analysis do you need to do to really be confident in your data? One of the things that happened to us is we've been lucky and appreciate TCEQ, Soil Board, and others for funding over the time. But one of the big problems we've had in this recent study was we were getting pieces of money to do pieces of projects. We weren't able to get funding to sit down and say, we want to look at ESP. We want to look at faecium in our sewage. We want to look at, you know, we want to do that baseline research that you really need to do before you can start saying we're going to be able to use these markers and this is effective and this is not. And it's very hard to get that funding from state agencies, particularly because it's driven by TMDLs and impairments. And so just a plea out there, it's in the long term, it's a really good investment to give researchers this funding up front to start to develop make good research, you know, have effective markers, have done all that baseline before you start to just go out there and try using markers. For our area, there's a lot more work we need to do with library-independent markers. Definitely want to move to qPCR because I think there's a lot of potential, and I do have a graduate student that is staying in Corpus. She's sitting in the back there, who's starting to work with some of the markers for different animal sources. And I'd like to acknowledge, like I said, a lot of different agencies have funded us: the General Land Office, Basin Estuaries Program, State Soil and Water Board. Jody's group, it's really great to be hooked in with that and to get that expertise through the GOMA money. So definitely thank you, Jody. Katrina's not here, but she was a really big keeper in terms of training the students and keeping us on track. Greg Buck, who's my colleague in college in Corpus, who's still there. Rick Hay, who is doing a lot of the work on Oso Creek with me. Roger and Michelle, who are the two master's students who are working on the ESP and the other molecular markers. And the environmental micro lab, which makes me feel a little sad now because this was my former life. Thank you all.